Okay, hello everybody. My name is Jessica Baker. I'm from UBC Okanagan, and the title of my research is Lavender Protects Against Infectious Colitis. So colitis is a type of inflammatory bowel disease, but unlike Crohn's disease, which can be anywhere in the gastrointestinal tract, colitis is specific for the large intestine. So it's a chronic relapsing immunologically mediated disorder in the gastrointestinal tract, and although it has an unknown cause, onset and activation is actually thought to develop in predisposed individuals. So in these individuals, the epithelial barrier is thought to be defective, so this allows for translocation of bacterial products into the lamina propria, and this triggers chronic activation and inflammation of the GI tract. So some symptoms of colitis can be very severe. You can get severe abdominal pain, fever and other signs of infection, severe weight loss. You can see this nice healthy colon here, opposed to this very ulcerative and inflamed colon here. And Canada actually has the highest incidence and prevalence in the world, and currently there are very limited therapeutic options. So to model this disease, we use a bacterium called Citrobacter rodentium. Now this is a mouse-specific intestinal pathogen. So this provides us with an ideal model to assess pathological responses in the large intestine with regards to bacterial-induced inflammation. So this leads to the production of various inflammatory mediators because the bacterium actually interacts with the surface epithelium, and this activates the immune system. So we get various inflammatory factors that are important in this model as well as in colitis, such as tumor necrosis factor alpha, interferon gamma, an enzyme, iNOS, or inducible nitric oxide synthase, interleukin 22, and macrophage inflammatory protein 2 alpha. Now these are all pro-inflammatory mediators, so they exacerbate the disease and cause damage, whereas interleukin 10 is actually an anti-inflammatory cytokine. So this one's good for it to be expressed, and it actually helps counteract excessive immunity. So this results in intestinal damage similar to human colitis. So we used a type of lavender called the Vandula X intermedia, cultivar Okanagan lavender. It was developed in a lab at UBC Okanagan by Sohail Mohammed. So they set out to improve the oil composition and the yield. So one of their main objectives was to decrease the concentration of linalool acetate because it's unstable and it's actually found in quite high concentrations in the wild type oil. As you can see, they were fairly successful in doing this, but they also greatly increased the concentration of cineol and borneol. Now, cineol and borneol have both been proven to have widespread anti-inflammatory properties, and cineol has also been shown to have antioxidant and antimicrobial properties, as well as an ability to inhibit pro-inflammatory cytokines such as tumor necrosis factor alpha, and borneol has been shown to inhibit oxidative damage. So we hypothesized that Okanagan lavender oil will decrease the severity of infectious colitis due to its ability to decrease inflammation and associated inflammatory mediators, as well as its ability to increase pathogen clearance. So C. rodentium infection is strain dependent. So for our procedure, we had two strains of mice. We had B6 mice and C3H mice. So B6 mice are susceptible to the infection, but they actually will recover, whereas C3H mice are highly susceptible to the infection, and it actually induces mortality. So these mice were orally gavaged mineral oil as our control, or lavender oil as our treatment, along with Citrobacter rodentium for 10 days, and we monitored their morbidity and mortality. So we collected the lower gastrointestinal tract, including the cecum and the distal colon, and these were put into RNA later, and this just preserves the tissue so we can extract the RNA for qPCR, or quantitative polymerase chain reaction. This just allows us to look at the level of gene expression of various inflammatory mediators. And we also H&E stained the tissue so we can score it for damage. We also collected the spleen, which filters the blood, and the mesenchymal lymph nodes, which filters our lymphatic fluid, to assess if the infection had gone systemic. So these graphs depict the morbidity and mortality of our two strains of mice. We have our resistant B6 mice along the top and our susceptible C3H mice on the bottom. So as expected, all of our B6 mice survived, but in the C3H mice, the lavender treated group showed a drastic decrease in mortality, with only 33% of our mice requiring euthanization on day 9 post-infection, opposed to 100% of our control mice requiring euthanization by day 7. And as a measure of morbidity, we were looking at body weight loss, So we didn't see a significant difference in our C3H mice in body weight loss, but in the lavender-treated B6 mice, we saw a significant decrease in body weight loss by day 10 post-infection. This is important because it's providing us with evidence that the lavender is actually protecting against wasting syndrome. So these are pictures of the cecums of the B6 mice. All these pictures are taken at the same magnification, so you can see the massive damage induced here. And you can see that the lavender-treated cecum actually bears much closer resemblance to our uninfected healthy cecum. So to properly assess and compare the extent of the damage, we use a method called histopathological scoring. So in this method, we look at 5 parameters. We look at edema, or infiltration of fluid beneath the skin, which you can see here. We look for infiltrating immune cells, so these purple dots here are infiltrating immune cells. We look for epithelial hyperplasia, which is the lengthening of the crypts, and this is a response from the host to trying to shed the bacteria off the surface. We look for goblet or mucus secreting cell depletion. So mucus is very protective, so those are important. And we look at epithelial damage. So we want to see a nice, smooth epithelial border, which you can see is completely destroyed here. So we assign a number to each one of these scores and add it up to get a final score. You can see our average final scores over here. You can see that the lavender-treated mice are showing a drastic decrease in damage to the intestinal tissue. So we have in vitro and in vivo evidence that the lavender oil can directly kill this pathogen. So in vitro, we have graphs depicting the colony-forming units recovered from the mesenchymal lymph nodes and the spleen. Notice that this is a log scale, so these differences are actually quite substantial. And this one mouse is almost completely recovered, which is extremely rare and significant. So this is important because it's providing evidence that the lavender may help with pathogen clearance as well as inhibiting a systemic infection. And then we have in vitro, we did microbial assays. We have a plate assay, so you can see our mineral oil plate is completely overgrown, whereas our lavender oil plate is showing quite a large area of inhibition. We did a broth microbial assay. So our bottom purple line here is a 10% lavender nutrient solution. The middle red line is a 10% mineral oil nutrient solution, and the top is just nutrient solution as our control. So these were inoculated with Citrobacter rodentium, and the optical density was monitored over a 24-hour period. And you can see that the lavender is directly inhibiting our microbial growth. So next we did qPCR. So this is to look at the level of gene expression of our various inflammatory mediators. So our pro-inflammatory mediators, which was the enzyme INOS, the pro-inflammatory cytokine, tumor necrosis factor alpha, interferon gamma, interleukin 22, and macrophage inflammatory protein 2 alpha. So you can see in the lavender-treated mice, the expression of these pro-inflammatory mediators is drastically decreased. And in our anti-inflammatory cytokine, interleukin 10, although a significant difference was not seen, there was a definite trend for it to be increased in our lavender-treated mice. So the mice that were orally gavaged with lavender oil are showing a drastic decrease in immune cell expression. So next we did immunofluorescence. This is used to visualize and specifically target targets with fluorescent dyes. So we were specifically targeting macrophages and neutrophils. Thank you. So we can see our macrophages are stained green, and you can see in the infected plus the mineral oil, there is quite a drastic infiltration of macrophages in the submucosal area here, whereas you can see they are absent in the infected plus Okanagan lavender mice. And our neutrophils, which are stained red, you can see there is, quite a drastic infiltration of immune cells here. You might also notice that the crypts are glowing red. This is because our marker for neutrophils actually cross-reacted with the bacterium, but it's fairly obvious that the infected plus Okanagan lavender are showing no infiltrating neutrophils in the submucosal area here. So the significant decrease in morbidity, cecal tissue damage, pro-inflammatory cytokine production, and immune cell infiltration provide strong support for our hypothesis that Okanagan lavender oil protects against infectious colitis. We're seeing protection via 2 mechanisms: an antimicrobial mechanism due to our microbial assays and our decreased pathogen load, as well as an immune modulation due to our decrease in infiltrating immune cells and our decreased expression of pro-inflammatory cytokines. So far our results warrant further analysis. We'd like to isolate and test the separate lavender components. We need to do more animal studies with a higher N number and varied dose amount. We need to examine overall health in other areas of the body, including the liver and the heart, just to ensure there's no cytotoxicity. And we can see two potential outcomes: an anti-inflammatory drug for chronic intestinal diseases such as colitis, or an antimicrobial drug for intestinal infectious disease. And I'd like to thank all of my coworkers and everyone with Merck for this great opportunity.